Wow.
So just an update for everybody. Welcome. We are going to get started on this webinar at the top of the hour. I will be talking to Dr. Nevan Krogan. Welcome to those of you who are logging on for today's webinar. We will get started at the top of the hour. If you have questions for Dr. Krogan, now is a great time to type them into the chat box. Hi, everyone, and welcome to our fifth but not final webinar focused on the COVID-19 pandemic. I'm Chris Rebelo. I'm the Director of Communications here at the Buck Institute for Research on Aging, and I have the honor of moderating today's conversation with Dr. Nevan Krogan, a molecular biologist who is leading a critically important international collaboration aimed at finding treatments for COVID-19. We hope that as much as possible, you are staying safe and healthy, mostly at home. As always, we encourage you to be an active participant in these sessions. You should see a chat widget at the bottom right of your screen. Please use that feature if you'd like to pose a question for Dr. Krogan. Nevan Krogan is a professor at UC San Francisco. He is the director of the intensely interdisciplinary Quantitative Biosciences Institute under the UCSF School of Pharmacy. He's also a senior investigator at the Gladstone Institute, and he is an adjunct professor here at the Buck. So here's background for today's session. Nevan's lab created a blueprint of the more than 300 human proteins that the SARS-CoV-2 virus requires in order to infect human cells and to replicate. Then he brought together an international research group to identify existing drugs and experimental compounds that target those human proteins, which means that there could possibly be drugs that would be repurposed to treat the coronavirus infection. More than 120 scientists have detailed the impact of 75 over-the-counter prescription and developmental stage drug compounds on the coronavirus. That research was published last week in Nature. Several of those agents shows promise in blocking SARS-CoV-2 replication in laboratory experiments we will talk about results. But first, Dr. Krogan, thanks so much for taking the time to be with us today. Before we get into the results, I want to talk about your approach to the work. So you are not so focused on the virus, you're focused on us. What's the advantage of that approach?
Yeah, hi. Thank you, Chris. It's a pleasure to be here. Thanks for the invitation. So as you rightly state, our focus is on the host. This is actually something that we've been pushing for some time now in terms of a strategy to help fight infectious disease. So there's a lot of groups out there that are trying to target the virus or viral proteins themselves, which is obviously a great strategy. You know, our approach is different. We're trying to identify the human proteins that the virus needs to infect us, and then let's pharmacologically target those human proteins. And there's advantages to that strategy. One, we don't mutate as fast as viruses. So one of the issues about having drugs that target viruses is that you get mutations and then you get resistance. And if you remember, the big breakthrough for HIV was actually a cocktail of 3 drugs that targeted 3 different viral proteins, and therefore that cocktail of 3 drugs was what actually overcame the resistance issue. Okay, so that's one plus, I would say, for host-directed therapy. Secondarily, we and others have shown that viruses, even very distinct viruses, are targeting similar human proteins. They're hijacking and rewiring them during the course of infection. So the logic would be, if you could actually have a host-directed therapy for COVID-19, it could be more easily brought to bear for COVID-22, COVID-24, or even other viruses that we're trying to combat as well. Obviously, there's downsides for host-directed therapy, and one of those is toxicity. However, in our approach, we're initially focusing on FDA-approved drugs and a lot of compounds in clinical trials that have already passed toxicity issues. All right, so that's our approach. And going forward, just like you saw with HIV, there may be a cocktail approach that's most successful. Maybe an antiviral like remdesivir combined with a host-directed drug or compound will ultimately be a great therapy in the future. But of course, more work is required to determine if that's the case.
So before we get into the results from your Nature paper, there is some breaking news this morning. Yeah, we talked earlier, you've been aware of it. There's some indications that the virus might be mutating and that there could be a more virulent form of the virus out there. Can you comment on that?
Yeah, I mean, this information has been available, I guess, now for a couple of months. I think it's a paper describing a mutation in one of the coronavirus proteins in spike. The question is that correlated with higher virulence? If there was an antibody against the original spike protein, would that antibody then also work on this mutated spike protein? The paper in question is actually getting a lot of press. However, to my understanding, it's a primarily computational paper, and there's a number of hypotheses, very interesting hypotheses. But I think much more data is needed to determine if this mutation is causing more problems, and if people have antibodies against the original spike, would it work on this mutated spike? So it's an interesting set of hypotheses, but more data is required to really test those hypotheses properly.
So there's no need for anybody to freak out about this?
Well, I guess everyone's freaking out already. So I would say, let's not freak out even more. I think this is an example of maybe the press latching onto something and maybe pushing it further than it should be pushed. But that's not to say these hypotheses, they could be real. But again, more data is required to actually prove it.
Got it. So let's talk about results from the research and the paper that was out in Nature last week. 2 drug categories have emerged as promising agents against COVID-19. One of the groups is called protein translation inhibitors. And the group that you're working with identified 2 cancer drugs that show promise against the infection. So first, what the heck are protein translation inhibitors, and how might a cancer drug work in this case?
Well, so there are a number of anti-cancer drugs that are out there that target the translational machinery. This is proteins in our cells that are needed to make other proteins. And this is obviously a set of proteins that the virus heavily relies on in order to make its own proteins. And in our map, we actually had identified a couple of key translational or set of translational proteins that we thought the virus could potentially need. And we identified a couple of drugs and compounds that target those specific proteins. One of them, we actually found a connection with this one translational factor that interacts with another one, that there is a compound that's targeting it. This is called Serpaphin, and it was from the company Effector. This is co-founded by a couple of UCSF scientists, Davide Ruggiero and Kevon Shokat. And we showed that it does actually, at least in a laboratory setting, has potent antiviral activity. This is a drug that's now being used in a clinical trial for multiple myeloma. So we're very excited about that. The company Effector is now looking to get this quickly into a clinical trial for COVID-19. And another protein, or that was a translational protein that was known to a drug or compound to target it. It was work done from Jack Taunton. Tenactin-4 is a compound that he had synthesized, and it targets the one protein EIF1A. And there's known that there's an FDA-approved drug. It's structurally distinct from Tenactin-4, but targets the same protein. And this is from a Spanish company. It's called Pladepsin, and they're actually have already started a clinical trial using this particular drug for COVID-19. So these are very potent molecules targeting key proteins in our cells. Toxicity is a concern, but for both of these, they've actually both passed toxicity tests, and we're very excited to see if they have any effect on COVID-19.
That's great. I want to back up for a second so folks get the big picture. When our cells are infected with COVID-19 or the coronavirus, what happens? What happens inside the cells?
So the virus itself is comprised of protein and an RNA, okay? And it injects its RNA into our cells during the course of infection, and it forces our machinery to make about 30 different proteins. There's only 30 proteins or probably less associated with the virus, and there's over 20,000 potential proteins in each one of our cells. So the virus cannot exist by itself. It can't live. It needs our cells, our genes, our proteins in order to live and replicate and infect our cells. So the way it does that is it hijacks and rewires our machinery, our proteins. So this was really the focus of the study is, well, let's try to systematically understand all the human proteins that the viral protein needs in order to make more proteins and replicate so that it can infect other cells. And during this process, as you alluded to earlier, we identified over 300 human proteins that we think are highly enriched for the virus needing in order to infect our cells. And then we looked at those to try to find drugs or compounds that would target those human proteins in a repurposing effort to see if they'd have any antiviral activity.
And then the cancer drugs, they act on the actual replication of the virus. I mean, they might block that replication that's happening.
It's not blocking the entry, so it's blocking the ability of the virus to make its own proteins.
Got it. Okay.
So you think of that as the replication process, if you will.
Got it. So the other category of drugs that your team identified are drugs that modulate proteins inside the cell. They're known as sigma-1 and sigma-2. Talk about that. Talk about some of the drugs that look promising in that category.
So, right. So we're very excited about this second category. And as you say, we're looking at two receptors, sigma-R1 and sigma-R2, both of which came out of the protein-protein interaction map as well. One interacted with NSP6, one of the cornevallar proteins, and another one interacted with another cornevallar protein, ORF9C. They're actually two sequence distinct proteins, but pharmacologically they're actually similar because similar sets of drugs and compounds are known to bind to and target both of these proteins. So we uncovered these, and working closely with Brian Shoykat, who's an expert at receptor biology, and he does a lot of chemoinformatics and makes predictions about drugs and compounds that bind to proteins, in particular receptors. So he's a perfect collaborator in this regard. And he identified a number of drugs and compounds that were known or thought to bind to sigma R1 and or sigma R2. And when we first looked at this, and we had reported 69 drugs and compounds, 4 of which we thought would target sigma R1 and R2, including actually hydroxychloroquine was one, haloperidol, an antipsychotic, and then 2 other preclinical compounds. And all of them had antiviral effects, which was exciting. And then that allowed Brian to augment the search. So then we included clemenstein, for example. There's antihistamines, other antipsychotics, which we're very excited about, also cough suppressants, and the female hormone progesterone as well. So we've identified actually a set of drugs or compounds that are related, but not at the chemical structural level. If you just look chemically at these, you would say this is not the same set of drugs. But if you know what the target is, and we think we do, and that being sigma R1 and R2, that's how you make the connection. And to me, this is why it's so exciting to start with the biology, then you go to the chemistry or the pharmacology. And then if you have something, you can go back to the biology. And that way, you're so many steps forward than if you're just randomly screening large libraries of drugs or compounds.
So I'm assuming you're not suggesting that anybody run out and start taking these drugs.
No, absolutely not. I mean, for us, it's kind of like a confirmation that these are receptors that are important. And keep in mind, obviously, they're just in laboratory setting, in cells, in monkey cells, African green monkey cells. So a lot more work is required. A lot more data needs to be collected to see if any of these drugs or these compounds would have any effect in humans. But I think we're on a particular area that shows a lot of promise. And Brian and the team are working very hard to identify new drugs that we're testing and essentially getting data every day on.
That's great. I just want to note John Newman, who's a geriatrician and a faculty here at the Buck. I talked to him on Monday, and then he asked me when I talked to you to just emphasize that antipsychotics and antihistamines are not good for older adults. So he just wanted to make that point clear. So people proceed with caution, which leads me to the question about.
Well, what I should say is they're not good. They're not good for anybody at high doses anyway. So I mean, there's problems across the board with them.
So let's talk about the cough suppressant that you looked at. That looks like it could boost infection. Talk about that.
So the drugs and compounds I alluded to with respect to binding sigma R1 and R2 are for the most part classified as antagonists, in that they're binding to these receptors and turning down their functions. And so Brian one morning told me, all right, if these are the right receptors, if we add an agonist, which turns up the function, we should see an increase of infection. And so that was a prediction he had made. And there was an agonist here for these receptors, dextromethorphan. And sure enough, in the laboratory setting, it did increase infection. So for us, it was really exciting from a scientific and biological point of view, because it's really helped us narrow in on it. We really think these are some key receptors for the virus. But of course, there's other implications here with dextromethorphan. This is a compound in pretty much every cough suppressant that's out there in the market right now. And obviously that's very ironic because, coughing is one of the major symptoms for COVID-19. But again, just like we talked about before with these other drugs, we're not saying that this drug is going to increase infection in people. However, we just wanted to responsibly report this information, and with a strong caveat that, look, we're just seeing this in a laboratory setting, obviously, and more data needs to be collected, not just in the laboratory setting, but in people and in humans as well. But we felt the need to try to, as responsibly as possible, report this, I think, very interesting connection to the public.
Got it. So let's talk about hydroxychloroquine. It's got a lot of attention in the media. Woo, this is going to be a great thing. And then there's questions. So I know you looked at it, and what's the status with that drug?
Well, we didn't want to look at it. The data told us to look at it. So this is really data-driven drug discovery. So, as I said, we generated this map of 300 human proteins, and unbiasedly made predictions about drugs and compounds, narrowed it down to 69. And sure enough, one of the 69 was hydroxychloroquine, which again, we weren't looking for that. That's what the data told us to look at. As I said, it predicted or to bind to sigma R1 and R2. And sure enough, it actually has fairly good antiviral activity in the laboratory setting. But it's a very dirty drug in that it hits multiple receptors. And in a collaboration with Brian Roth at University of North Carolina, who's one of the best in the business at studying these receptors, he did some very interesting in vitro analysis for us, comparing the affinity of hydroxychloroquine, its binding to sigma R1 and R2, compared to hERG, the receptor for the heart. If you remember, there's a lot of hydroxychloroquine clinical trials ongoing, including one that was in Brazil, which is a very big one. It was stopped because mostly for cardiotoxicity issues, problems with the heart. Well, the data that Brian generated for us nicely showed, again, in an in vitro setting, that hydroxychloroquine binds much more tightly to the hERG receptor that's associated with the heart cells than with sigma R1, compared to a lot of the other drugs and compounds that we've uncovered. Okay, so our prediction, at least in the laboratory setting, is that some of these other drugs and compounds would not have that same cardiotoxicity issues if it was introduced into people. There may be other issues, there may be other toxicity issues we're just not aware of, but at least with respect to the cardiotoxicity, we don't think we would see an issue there. So we've generated some molecular data that I think helps support why hydroxychloroquine may not be the best drug for COVID-19.
And you found another drug that looks even much more potent than that drug, right?
We found it's actually a compound. It's a preclinical compound called PB28. It was originally synthesized and studied in the 90s. I think it was going to be, it was being looked at as a potential antipsychotic compound. And in the laboratory setting, it's actually 20 times more potent than hydroxychloroquine with respect to its antiviral activity. And as I said before, it's not binding as tightly to the hERG receptor associated with the heart. It has a much higher affinity for sigma R1 and sigma R2 compared to hERG. However, this is not an FDA-approved drug. We're actually looking now to get this into mammalian models to look at its antiviral effects and potential toxicity. But it's super exciting because we've identified a class of compounds and their proposed targets. So now people like Brian can look at this and start to identify many more compounds and drugs. We've actually got several new FDA-approved drugs in this category that we've been testing, and they look even potentially stronger than this one preclinical compound with respect to having antiviral activity. So we're really excited that we've identified this set of drugs and compounds. And although this one will take a long time to get into people, there's some other ones that are already in people that we think will be just as good as this preclinical compound, and we're very excited about that.
So what's happening now? Like, what's the state of the research?
Well, we have, obviously generated this map looking at, SARS-2 in one cell type. We've made predictions about drugs and compounds. We've been collaborating with scientists in two different continents, two fantastic virological institutes, the Department of Microbiology at Mount Sinai in New York and the Institut Pasteur in Paris. There's some great work going on now in San Francisco to get the virus up and going, including, Melanie Ott at the Gladstone Institute, who's also an adjunct faculty at the Buck Institute. I think she's got this up and running. We're going to be working with her to test some of these. But out of the 69 drugs, we actually only really looked closely at 47, only 2/3 of them. So we're continuing to screen the other third. And when we get hits, then we can open up chemical space to look at other drugs and compounds like I had talked about before. There's a lot of effort now to get some of these, especially obviously the FDA approved drugs, into clinical trials, or these compounds that are already in clinical trials for cancer, like multiple myeloma, like these translation inhibitors that I talked about previously. So we're focusing on how best to get these into people to see if they actually have anti-COVID-19 activity. But then I should say the map itself is spurning out a lot of hypothesis-driven research. We've actually started up this, it's called QCRG, the QBI COVID-19 Research Group, at UCSF. And actually, it also at the Buck Institute, it harbors Eric Verdun as well, the president of the Buck Institute. And it started with 22 different labs, and now it's expanded to over 40 labs, essentially encompassing hundreds of different scientists in each one. If you look at the accumulation of all the people in the lab, and it's gotten so big, we've got these different subgroups. We've got 10 different subgroups focused on different technologies like structural biology or bioinformatics, and then specific biological areas too, like translation and ubiquitination and so forth. So there's a lot of work ongoing, characterizing these interactions in a more deep fashion, trying to really mechanistically understand a lot of these potential connections that were uncovered. And for me, this is a really unprecedented collaboration. I've never seen this many scientists come together and work together so closely on one particular problem. So for me, that's very exciting. But then also to say, we're in the process of generating more maps. So we looked at SARS-2, we got SARS-1 genes cloned out. We want to do a comparison there. We're going to do MERS. We're going to do another coronavirus that's not as virulent like OC43. We looked in one cell type. We want to look across other cell types, and then also in bat cells as well. So there's a lot of discovery research that still needs to be done. So that's really exciting to me. We got a lot of chemical biology approaches that need to be done in terms of trying to find best of class compound in these different classes. And then there's a lot of work trying to figure out how to translate these findings and more into the clinical world. All right. So there's so much more work to do. So we're going to obviously be kept busy for a long period of time.
Do you have any idea of timeline? So like, let's say for the drugs that you talked about, some things are already in clinical trials. What's the timeline for possibly going, yes, this works, this drug is available?
Yeah, so obviously these clinical trials take several months. I mean, I think we'll see. I mean, the epidemiologists are predicting that we're going to see a lull in infections in the summer and then a spike in the fall. And this is what we saw with the 1918 flu epidemic. And I think there's no reason to believe that that won't happen in this case as well. So, I mean, maybe best case scenario, 6-7 months with some of these drugs, if the trials could get done, maybe they could be brought to bear to help in the fall when the second wave of this pandemic hits. So, we're trying to raise money to help push these things through. So there's a lot of things connected here. Obviously, a lot of people have helped us. I got to mention one person, Ron Conway. He's been, I think he's called the father of the Silicon Valley. He's been helping us raise funding, and he's been kind of a hero here, helping us. So we have people helping us raise money to push the science forward, but there's still a lot of work to do. And probably the best case scenario is if one of these could be brought to bear maybe by the end of the year.
Okay, that's great. So whenever I do these seminars, I always get a lot of questions from folks who want to know about natural products that are thought to boost the immune response. I got a question this morning about quercetin. I get questions about vitamin D, vitamin C, lots of stuff. Is there any way at some point, is there interest in those? How is your work, could that eventually validate or not validate the compounds that folks are told to take to boost their immune function?
Well, I think obviously the stronger your immune system is, right? The more effective you will be at fighting off not just COVID-19, but other infectious agents. But my guess would be a lot of those would be, in that case, indirect effects of you fighting off infection. And what we're trying to do is to get, using mechanistic approaches, data-driven approaches, trying to get right to the heart of the infection. And this is why we're looking specifically at human proteins directly touching these viral proteins and trying to then pharmacologically target those human proteins that are presumably much more direct than maybe some of these other connections. So that's the way we're looking at it. We're trying to get as mechanistic as possible using biology, mechanistic biology, to help drive our experiments. But of course, there's a lot of other approaches out there. And, my gut feeling is probably some combinatorial approach will be brought to bear on this, just like with HIV. You had a cocktail of 3 different drugs at the end of the day. That was the big breakthrough. So one could envision maybe one host factor, and as we said, remdesivir, combining that, or a couple of drugs targeting the virus, or a couple of drugs targeting host proteins, or some combination of that. So I'm hopeful that approach, which has worked in the past on other viruses, could be brought to bear here.
So do you have folks who are actually working on designing new drugs based on some of the findings that you've got when you've talked about drugs that maybe have toxicity problems? Or
Absolutely. So this is Kayvon Shokat's probably the best in the world at this, and he's as is Brian Shoykat's getting involved, and we have other people at UCSF like Matt Jacobson and Jack Taunton, and other people around the world, obviously. So once you have this chemical matter, and what the target is, that's important as well. You can be so much more powerful in terms of tinkering around with these drugs, studying how they interact with their specific targets. And then in a reiterative way, you can design something that has a higher affinity and hopefully is much more potent. So that's really the power, in my opinion, of the approach we're taking. Is it's really data driven and leveraging such great expertise that exists in the Bay Area around chemical biology. We got the best in the world in that, and they're all working together in a really exciting way, in my opinion.
so I want to talk about the work and the collaboration. One of the things I find most heartening about what's going on is it seems like the scientists are collaborating, they're cooperating, they're not competing. International boundaries don't matter. Like, let's tackle this. So can you, like, how often are you on Zoom meetings with people from all over the place? What's your day like?
Well, so one thing I'll comment on initially is your point of collaboration, which I think, just like you, I think it's been absolutely fantastic. And one of the problems in my mind with science is that it's very siloed and very competitive, unnecessarily competitive, in my opinion. And it's competitive because the structure is set up to really reward the individual. The one person gets the award, one person gets the grant, one person gets tenure. And this structure often discourages collaboration, especially for young scientists who have the best ideas or the most enthusiastic. They become more protective because of the way the system is set up. But here, what we've seen is these silos have really been broken down. And I think as a scientific community, not just with us, but around the world, we see how fast science can actually move. And breaking down silos across different laboratories, across different institutions, and then also between academia and pharmaceutical companies. We've seen this as well with us and with other groups. And for me, this has been really the most exciting part of this. And if you just think about it, this is a virus we didn't even know about really at Christmas. And now here we are at the beginning of May, 4 months later, saying, all right, we cloned it, we generated a map, we made predictions about drugs and compounds. We got the virological assays up and running. We've tested these. We've got some chemical matter that we're trying to get into humans now in the matter of less than 4 months. And to me, that's unbelievable. And the question is, why can't we be doing this more often? And the challenge now is to sustain this infrastructure that's in place by changing it, changing the rules of the game in many ways, hopefully, so that we're in a much better position when COVID-22 or COVID-24 hits, or we just want to study another virus or just study any other disease like breast cancer or Parkinson's. Why aren't we doing this, studying all diseases? So for me, this has been a real silver lining here, is really seeing how fast science can move. And I would say that the Quantitative Biosciences Institute, this is really what we've been about the last 4 years, is trying to break down these silos on so many levels. So in that regard, I would argue we were in a perfect position to respond to this pandemic. We've been seeding these collaborations, both in the Bay Area and around the world and with pharmaceutical companies. So we were in a really great position to act on this particular pandemic. And to your last part of your question about Zoom, yeah, I mean, it's, there's been some meetings where we've had several hundred people on Zoom. That just becomes unwieldy. It's kind of cool to see with respect to this QCRG. But now we've got these 10 different subgroups. So now they each have like 50 or 60 scientists associated with each subgroup. And the question is, when the dust settles on COVID-19, how much more are we gonna be doing Zoom? I did a thesis exam 2 days ago in San Diego, and I was the external examiner, and it took an hour and a half, and then I was done, and that basically saved me a day and a half of travel. So the question is, how much Zoom are we going to be doing in the future? Probably a lot more than we've been doing in the past.
So I want to talk about some of the work that's happening here at the Buck. You mentioned Eric Verdon's lab. So they're looking at aging and metabolism. So can you talk about that? Like what happens metabolically during an infection? What happens inside cells?
So one of the subgroups associated with QCRG is this aging metabolism group, like you say, and that's being run by Eric and also Natalia Jura at UCSF. And obviously metabolism is a key process that's being perturbed in the context of infection, and is also, I think, the virus is more actively hijacking it as well for its own benefit. And that's, I think, nicely reflected in the protein-protein interaction map that we had generated. There's several metabolism enzymes in there that look incredibly exciting, many of which are connecting to aging. There's a lot of things obviously connected to aging. And there's some enzymes in there that Eric has worked on. I think he's discovered some of these, many, many years ago. He doesn't look that old, but he's been around a long time, and he's discovered a lot of these enzymes, and he's in the perfect position to characterize these. And it's been a great pleasure interacting with Eric and a number of scientists at the Buck Institute. And again, it's a testament to the collaborative relationships that have been formed, in the Bay Area around this. And the challenge now is to keep the momentum going, not just on COVID-19, but other diseases as well.
That's great. I got a question, and somebody's asking about antibody tests. And, there's a big desire for those. Is your work, I know right now you're targeting the infection, but your approach, would that be useful in the future for looking at antibodies to infections?
Well, there's a lot of efforts to get antibodies, not just for testing, but for treatment. And in the context of the QCRG, there's a lot of effort around that. A lot of work being done by Jim Wells and Charlie Craik and others, trying to get at these antibodies, as I said, for diagnostic purposes and hopefully longer term treatment purposes. But if, again, back to this idea of understanding, the more you understand about the biology of the virus, the more powerful you are with respect to treatments, therapeutics, and diagnostics. So if there's a potential viral protein associated with a human protein, having an antibody that would recognize both in the future could be much more powerful. So again, it's this idea, the more biology you know, the more powerful you are going forward. And that's really the message of all discovery research, in my opinion, and how you can translate it into something therapeutic.
Got it. So were you surprised at the hits that you got? Cancer drugs, antihistamines, progesterone. It's like they're all over the map. Were you surprised by that?
So we've generated a number of maps in the past targeting many other viruses, including HIV and dengue and Ebola and Zika, hepatitis C, influenza. And it's really cool to look at the biology that comes out of these maps in general, right? Because these viruses, they rely on our machinery, as we talked about before, and they're going to be targeting key nodes and key proteins and key complexes, and essentially a lot of major biological processes in our cell. It's this virus, this SARS-2, in terms of the richness of the map, I haven't seen anything like it. And to me, it's actually quite exciting. It's really getting its fingers in a lot of different areas of biology of our cell, in a way that I've never seen before, as we looked across, 20 other viruses using this particular approach. So in that regard, it's not such a big surprise that we're, since it's across many different areas of biology, we're getting a lot of different drugs and compounds encompassing that wide breadth of biology. But what I'd also just say too, and we've reported on this in the past, is you see these commonalities as you go across different viruses. You see the same proteins being hijacked and rewired as you go across, RNA viruses, DNA viruses, lentiviruses. But if you take a bigger step back, we're also using these approaches on a variety of other diseases like cancer and neurodegenerative disease and heart disease. You actually see amazing commonality there as well. It's the same genes being hijacked by HIV that are being mutated in cancer. It's the same genes being mutated in Parkinson's that are being hijacked by Zika. And to me, that makes sense. You got these Achilles heels of the cell, they become mutated and they result in disease X, Y, or Z, or a virus has evolved to attack these. And that ultimately results in disease. And why don't we see this? It comes back to this problem of silence. Science is that we're too siloed, is that people just focus on their one disease, and they can't recognize these commonalities. Well, our approach allows us to scan across these diseases and find these commonalities. So then it's not such a big surprise in my mind. Well, an anti-cancer drug would actually could be used to fight a virus, or vice versa. Maybe there's an antiviral out there that could be helped to fight cancer. So if you just think about it from a biological point of view, and you start to collect this data across these disease areas, to me, it actually makes sense how one drug or treatment for one disease could be brought to bear for another, or vice versa.
Got it. So people talk about viral load, like healthcare workers who are around people who have COVID-19 can get a really big viral load. And other folks, if you're just walking down the street and you're walking by somebody who has COVID-19, you might not get such a large viral load. Is your team looking at that, or is that going to be something for clinical trials? Can you talk about that?
Well, we always try to get to the molecular mechanisms behind these phenomena, right? So to me, it is really fascinating as you're talking about why certain groups of individuals are seemingly very susceptible to this virus, and others, there's no symptoms at all. They're completely asymptomatic, and probably the vast majority of people are going to fall in that latter category. So the questions are, well, what separates out these individuals? Clearly, there's other health issues that contribute, but there's a lot of just seemingly very healthy people, younger people, that fall in either one or the other. And the question is why? Well, there's a lot of genomic analysis being carried out right now, sequencing analysis that's being generated, and there's signal that's being looked at in the DNA. So people are looking for rare variants, like mutations, if you will, in one group versus the other, and identifying genes that could be contributing to the susceptibility of these resistance. And that's the kind of information that we would love to have, because that map would help us interpret that genomic data. So if there's a mutation in one of these host genes that the virus needs, we can then study it in a very detailed way, biochemically, biophysically, and structurally, and really try to understand the underlying biology behind some of these molecular connections and what these particular variants are doing. And in fact, this is what we've been doing in other infectious diseases like HIV. We've generated this map with like HIV. Then we've looked for these people that are resistant to HIV, these highly exposed seronegatives. This is a collaboration with the group in Chicago. They've identified several genes and variants in these individuals that then we go and study at the molecular level. So really the goal in my mind, at least from our work, is to try to connect our data with the genomic information from people that are either being very resistant or very susceptible to the virus, to get a better understanding of the underlying biology, which then hopefully would point in novel therapeutic directions in the future.
Got it. So you work with this stuff? You're trying to fight this virus? People are talking about, is it okay to go outside in a t-shirt? Should I take showers? I know that's not your area of expertise. Personally, are you a germaphobe, or what? How do you approach everything that's going on a personal basis?
Well, yeah, I mean, as you point out, that isn't my area of expertise. And actually, in the scientific community, we're trying to learn about this virus. It's like every day we learn something new. Me personally, I mean, I have started to go back to work. There's not a lot of people at work. There's still a shutdown at UCSF, and only those that are focused on COVID-19 research are allowed in. And there's a lot of social distancing rules in place as they should be. But what concerns me the most is, I hear people talk about this, even some of my friends that aren't scientists, saying, well, I don't know, maybe we can start to, start up life again. And to me, that kind of talk is very scary. And that type of behavior will ultimately lead to a spike, and that spike could be even worse than what we've seen so far at the end of the day. So, my gut feeling is, and there's obviously better people to speak on this area, but my gut feeling is we need to be very vigilant here in the social distancing rules that have been put into place. And I think we should still be hunkering down for a lot longer than some of these people are talking about.
So personally, I agree. And also tests are starting to come out. They're starting to become more available, but it seems like testing is crucial to how we move through this thing.
Yeah, I completely agree. We don't have the testing capability to kind of re-engage society. That's what we need, and testing, not just to say who have been infected, but then serological tests that to say who has potentially been infected and who have recovered, right? Until we have that wider range of testing capabilities, I think it's even more reason to stay shelter in place, at least in the short term.
I got it. So as your group, are you working with biotech companies, big pharma? Can you talk about the positive fallout from what you're doing and how other companies and organizations are picking up the work and carrying it forward?
Sure, that's been another really exciting aspect of what's gone on over the last few months, not just with our work, but we see this with other people's work around the world. And I alluded to some of these connections. So, for example, the company Effector, which has made this one anti-translation drug, which seemingly has strong antiviral activity in the laboratory. Now they're starting to push that compound into a clinical trial. So it's been great to interface with them. The Spanish company, which has this pledepsin, which targets another translational factor, that's going forward, and it's great to be interfacing with them as well. Also with the pharmaceutical company Roche, we'd actually been working with them over the last couple of years on a couple of other respiratory viruses. And we're interfacing with them right now. They're providing some funding for us to do work around this idea of coming up with host-directed therapies. And now we're in close talks with them to see if they can help us translate some of our findings into the clinical setting. And also, I'd just like to say, other companies, smaller companies, a lot of them in the Bay Area have approached us and have offered to generate reagents for us, to give us reagents free to see how they can help out. And there's been really a lot of generous behavior from, small, medium-sized companies, not just in the Bay Area, but around the world, trying to help us out and help other academic groups out as well. And as I said before, the goal is to keep these bridges strong across the academia and companies, large and small, not just for the foreseeable future to fight COVID-19, but hopefully these are bridges that'll stay in place as we tackle other diseases in the future. Because both sides have so much to bring to the table, and if you can put this together, we're so much better off, I would argue.
So how about, we talked about this when I chatted with Larry Brilliant a couple weeks ago, future pandemics. I think the Trump administration has axed funding for a project in China that was going to look at bats to see what else is brewing in bats. You've looked at lots of viruses. What's out there? Like, what you're getting to the source of what's happening in the cells. Do we need to get to the source of what's causing these viruses to show up in humans anyway?
Yeah, I completely agree. And it's actually, if you think about it, it's not that big of a surprise we're dealing with COVID-19. If you look at SARS-1, that was back in 2002, 2003. That had a higher mortality rate, but it wasn't as infectious, and that got under control, which was obviously great. And then there was MERS, another coronavirus, that was in 2012. Again, even a higher mortality rate than SARS-1, but again, not as infectious as SARS-2, and that was contained. And the idea there was it went from a bat to a camel and into humans. And now, a few years later, we're dealing with another coronavirus. And I think the logic there was a bat to a human, or a bat to a pangolin to a human. So I couldn't agree more, studying what could be the next pandemic, going to these different animals to study them. One of the best in the world at studying zoonosis at a molecular level, at least, is Harmeet Malik at the Hutch in Seattle. And we're working very closely with him. He's making some predictions about viruses that are presently in bats, and pointing us in that direction in terms of generating some, molecular maps based on those viruses. But that also has to come from funding agencies, So I think one of the most powerful ways to study viruses is to study them in an evolutionary way. And we've worked on HIV for years. And we, in humans, obviously, but we really wanted to extend that to SIV in primates, and then there's FIV feline, there's MMV in sheep. We tried to get money from NIH. They said, no, if you should only study HIV in humans. There's no point in studying these other viruses and other species, which I couldn't believe they said this. So I think that the funding agencies have to have a more of an open mind to provide funding for us to study in these other species, because this is how these viruses are coming to us, through these other species. So let's try to understand at the molecular level what's happening with these viruses in these other species, and then that'll give us a better idea of when something's going to jump, and then hopefully it'll point to more powerful ways for to come up with therapeutics or diagnostics. So I couldn't agree more. We need more funding to study these viruses and be more predictive of when the next one's going to jump to us.
So I know there are other groups working on vaccines. That's not your specialty. I read something this morning in the news where, predictions that we might have something by the fall, probably, even if they find something that looks good, to get it tested, to make it available is not happening. What do you, if you were gonna, put your, look at your globe to predict the future, do you think it will be a combination of vaccine and treatments? I know in older adults, vaccines often don't work as well. Like, what do you think this is going to look like once we get a handle on what to do with this virus?
On one hand, I pray there's a vaccine as soon as possible. And then you think, well, there's a lot of viruses out there where a lot of people have spent effort trying to get vaccines, and they haven't, viruses that have been around for decades. But then on the other hand, I don't think you've ever seen a situation where so many scientists and great minds are really solely focused on trying to solve this problem, especially in the context of the vaccine. So I just don't know. I really hope something, something does come up with respect to a vaccine, but you just don't know. That's why you got to be pushing on all fronts, right? So therapeutically, obviously, we're focused more on drugs and the pharmacological approach. There's also a lot of work with respect to identifying antibodies that could be used therapeutically. So I think we need to push as hard as we can on all fronts. And, as you said, and as I talked about previous, at the end of the day, it's probably going to be a mixture of approaches that'll be brought to bear to help out here. And it could be, with respect to therapeutic approaches, maybe a drug and an antibody or something, or as you say, maybe there'll be a vaccine for a certain subgroup and there'll be a therapeutic for another subgroup of individuals that are older or don't respond to the vaccine. So that's why it's so important to keep pushing on the research on our fronts, because you don't know what's going to break out and be the most useful.
So I'm getting some questions about COVID-19, and it looks like in some cases it might induce thrombosis. There's been issues with blood clotting. We've talked about issues with the heart. There's been some indications that the virus can impact the central nervous system. So I know your basic research, is it going to help explain sort of the other effects? The initial thing was, oh, it's all about respiratory. It's respiratory, which it is, but it's not just respiratory. So is your work informing that too?
Yeah, absolutely. That's the vision here. And as you point out, this is a virus that the likes of which we've never seen before. And I was actually just talking this morning to Adolfo Garcia Sastre, one of the best virologists in the world in New York, and he was just lamenting the fact that this manifests itself in so many different ways, in ways we probably don't even understand yet, as we do some of these retrospective analyses. So I think it's going to be so important to study at a molecular level what this virus is doing from different tissues, from different organs, and why, and then combine that with genomic data from the individuals. Why with some people are they going to have certain symptoms and other people will have other symptoms, looking the same age and the seemingly similar health situation. So I think the more molecular data we can collect around these issues, the better understanding we'll have, and then the more predictive we'll be going forward.
That's great. We're almost at an end here. Is there anything that I haven't asked you that you think it's important for people to know about this virus, the work that you're doing?
I would say we kind of touched upon this with respect to funding. We're always struggling to find funding, either through federal means or through philanthropic avenues, And it's so important, especially in times like this, to have flexible funding, because none of us have grants on COVID-19, because it takes, months, if not years, to get grants in. And we've all shifted our focus, or a lot of us have shifted our focus to study COVID-19. So we need to increase funding across the board in every way possible for discovery research. So that's one thing. So we're in a better position in the future to tackle the next pandemic. And then, as we talked about before, I think it's very near and dear to my heart, is how do we keep this collaborative infrastructure in place so that science can move this fast in the future. And it really, again, comes down to breaking down these silos, making these connections, maybe reconfiguring the reward system that we're all dealing with on multiple different levels, so that we can have this new way of doing science, this new paradigm set up more effectively in the future. And that's connected to funding as well, obviously, so it's all connected. But I've just been so really delighted to see how people have been working together, and the challenge now is to keep that excitement and that level of collaboration going forward for the next several decades.
Got it. Thank you so much for making the time to be with us today. I want you to get back to your important work. You're a hero, in my opinion, so thank you very, very much for being with us.
Thank you very much, Chris, for the discussion. It was a pleasure to be here.
Great. So I want to thank everyone who joined us today on the call. A video of today's event should be posted sometime tomorrow. All of the videos from our previous webinars are also posted for you and your friends to watch at your leisure. So today marks the last webinar in our first series of webinars focused on COVID-19. I am here to tell you that we will be back with more. This pandemic is far from over, and we want to continue bringing you evidence-based information on the status of the pandemic and the science aimed at bringing it to an end. We haven't finalized our schedule. Be assured that as soon as we know, you'll know. In the meantime, be sure to visit our website for the latest news. You can also sign up for our newsletter. And if you found value in these webinars and following up on what Nevan just said, I ask you to please consider supporting our COVID-19 research. Options for donating are on the support section of our website. We are, many labs here at the Buck have pivoted to include COVID-19, and we really could use your help. So for now, thank you so very, very much for participating in these webinars. I have so enjoyed doing them with you. Thanks for your questions, and thanks for caring enough to let us into your life. So please be healthy, because I plan on seeing you back in this space in the very near future. Bye-bye.